Yes, we are in our last session from Microbial Diversity, where we are going to deal in detail about viruses today and going to practice objective. Fine, right? So let's get started for a strong objective session and complete information of viruses. Viruses we are going to learn its structure, replication, viral diseases transmission methods, and plant viral diseases, especially tobacco mosaic virus and rice turgovirus. First, if you see, the discovery of virus is very important. Edward Jenner is the first scientist in the year 1798. He has isolated, he has successfully vaccinated against smallpox using a material from cowpox lesions. Okay, using a material from cowpox lesions. Next, it is Louis Pasteur. He developed a vaccine against rabies in the year 1818. In 19th century, harmful agents are referred as viruses. They are referred as viruses. This is derived from Latin word which means poison. Charles Chamberlain, he is a collaborator of Pasteur. He demonstrated the rabies disease agents were filterable. That led to the discovery of virus as what? Filterable viruses. They can easily be filtered through bacterial filter paper. So they are addressed as what? Filterable viruses. Now comes 16th century. In the 16th century, plants were also infected by viruses. Charles has reported one virus, that is tulip break virus. This is one virus. Find it. Who reported that is important. Next, it is the scientist Bejering who observed the symptoms in tobacco mosaic virus, and he said that the disease is being transferred from a infected plant to a healthy plant. The fluid which is extracted from the infected plant is affecting the healthy plant also, which is addressed under the term contagium vivum fluidum. This is reported by Humma Bejering, okay, by the scientist Bejering, who has observed tobacco mosaic virus, who has observed the disease tobacco mosaic disease from the virus tobacco mosaic. Fine, right? Now if we see few more scientists and their related discoveries with respect to viruses, it is Loeffler and Frost who has discovered foot and mouth disease in cattle, which is caused by filterable virus again. Next, Walter Reed in the year 1900, he revealed that yellow fever in humans is caused by filterable virus. What is the vector that is transmitting this viral infection? Mosquitoes. Fine, right? Next, Twart in the year 1915, he isolated bacterial viruses, followed by D. Herrle in the year 1917 discovered what? Bacteriophages. Bacteriophages are those viruses which target and infect bacteria. Fine, right? Now, the process of bacterial destruction by viruses is referred to as Twart-D Herrle phenomenon. Okay, this is addressed as or referred as what? Twart-D Herrle phenomena. Next, Schiller separated what? WLL bacteriophage using ultracentrifugation method. He used ultracentrifugation method identifying the presence of protein and phosphoric acid, which shows viruses exist in crystalline form. Viruses exist in crystalline form. That is said by identified the presence of Schiller has identified the presence of proteins and phosphoric acid, showing that viruses can exist in crystalline form. And it is W.M. Stanley who has first time crystallized viruses. He has crystallized TMV and he got a Nobel Prize for that in the year 1949, which is an important discovery. W.M. Stanley. After Bejering, this is one of the important discovery. Next, Bavden and Pyrie, he succeeded in separating what? TMV virus particles into protein, recognizing that viruses are unique organisms with pathogenic abilities. These first two introductory slides will give you about the discoveries, wide range of discoveries of viruses, where the information about the discoveries is very important. Mostly the questions regarding discoveries will be asked in the form of match the following type. So, one or two revisions regarding viral discoveries is important for you people. Next, here comes the characteristic features of viruses. Viruses have many synonyms, like they are addressed as infectious agents, or acellular pathogenic organisms, they are addressed as infectious agents or living entities or acellular pathogenic organisms. Virus will just have genetic material and a protein coat over it. Okay, what are the components of virus? Virus will have a genetic material. That genetic material can be either DNA or RNA based on the mode of infection. And second is a protein coat. Okay, a protein coat. In few viruses, above the protein coat, envelope is present. Okay, envelope is present above the protein coat in few viruses. Genetic material and protein coat over it, followed by it has an envelope. Fine, right? Enveloped viruses. Only few viruses are enveloped viruses. These are the components of a virus. There is no particular cell or cellular constituents that are present. That is the reason they are addressed as acellular pathogenic organisms. Clear till here, ma? Any doubt anywhere? Yeah. Now, the study of viruses is known as virology, and the scientists who are studying this viruses are addressed as virologists. Viruses are unique living organisms. They lack basic physiological functions and cell structures are lacked. Non-living outside the host. Viruses are non-living outside the host, whereas when they enter into the host organism, they replicate fast inside the living host. Important point to be addressed. Next, they are capable of inheritance and mutation like living organisms, but this capability of inheritance will happen only when they enter into the host cell. Without host cell, their machinery is zero. Hence, viruses are considered as link between living and non-living organisms. Clear till here, ma? Now, next, structure of viruses. General structure, if you see, a complete virus particle is known as virion. Okay, complete virus particle is virion. It has two components. One is genetic material. It can be either DNA or RNA, and a protein coat. Protein coat is known as capsid. So, together along with the genetic material and protein coat, you can also address it as nucleocapsid. Okay, you can also address it as nucleocapsid. The nucleic acid is enclosed by a protein coat, that is capsid. Sometimes it also has an additional layers which may contain carbohydrates, lipids and proteins. Okay, virus particles like virions, they range from 10 to 400 nanometers in diameter. Okay. Next is genetic material in viruses. It is always nucleic acid. It can be either DNA or RNA, but never both in a single virus. Fine, right? Plant viruses will have RNA, animal viruses DNA, and bacterial viruses also has the genetic material, that is DNA. Now, exception. Cauliflower mosaic virus is an exception. Exceptions are always important, ma. Exception like cauliflower mosaic virus, this has DNA. Fine, right? Influenza virus or F2 colifisis, this also have RNA. Now, animal viruses like RNA can be either single stranded or double stranded. Viruses are acellular, that we know, and they do not undergo any cell division or reproduction in the absence of host cell. Clear? Now, protein coat or capsid. They gives virus a definite shape and antigenic properties. They will give a definite shape and antigenic properties. Capsid is made up of protein subunits called as what? Promoters. Capsid is composed of a single type of protein and multiple types of proteins. They can be of three symmetries. It can be helical, icosahedral and complex viruses. Viruses with an envelope is called as enveloped viruses. Viruses without any envelope are known as naked viruses. All this is clear, right, about the genetic material and protein coat? Any doubt anywhere, ma? Yeah. Now, examples of capsid viruses. We have three types as we have discussed: helical, icosahedral and polyhedral. Helical viruses examples include TMV, where the capsomeres here. Capsomeres are what? Protein subunits. The entire protein is addressed as what? Protein coat is capsid, and in the protein coat the protein subunits which are present are known as capsomeres. These are protein subunits. They are spirally or helically arranged, forming what? Long rigid tube. In icosahedral viruses, for example, adenoviruses, capsids are what shape? Polyhedral shape. Next, in bacteriophages, the head is icosahedral with overall shape is complex. Next, enveloped viruses. What is about envelope? Envelope will infect mammals. Special example I can, except smallpox viruses, all the viruses which are infecting mammals will have an envelope, which are addressed as enveloped viruses. These are often referred to as lipoviruses as well. Okay, the envelope derived from what? Nuclear or cytoplasmic membrane of host during viral release. This layer converts into an envelope containing peplomere subunits. They contain what subunits ma? Peplomere subunits. Enzymes found in viruses include lysozyme in bacteriophage, neuraminidase in influenza. Enzymes are very important because these enzymes will dilute the host cell wall layers. Okay. One is lysozyme in bacteriophage, that will degrade the bacterial cell wall peptidoglycan. Okay. Lysozyme enzyme will degrade or degenerate the cell wall, that is peptidoglycan, which is present in the bacterial cell wall. Neuraminidase in influenza virus, fine, right? This also includes RNA polymerase and reverse transcriptase enzymes as well, fine, right? This is about capsid and envelope. Here we go with, of course, we have already dealt with the examples of helical, polyhedral and complex viruses. Here we go with the detailed note on each type of virus. Clear, right? Now we see helical viruses. Most plant viruses are helical or rod shape or elongated. One best example I can give you is TMV. Here for tobacco mosaic virus, it is important to note the length. Length of tobacco mosaic virus is around 3000 angstroms, that is 300 nanometers in length. Diameter is around 180 to 190 degrees. Okay, Armstrongs diameter, and central core form a hollow tube at the center, which is of 40 angstroms diameter. Okay, hollow tube at the center is of 40 angstroms diameter. Fine, right? This opens at both the ends, 3.5 to 4 micrometers. Capsid is made up of, capsid, that is protein coat, it is made up of 2130 protomeres, sorry. Okay, 2130 protomeres, or you can also say capsomeres. Capsid is the protein coat completely. It has protein subunits called as protomeres or capsomeres. How many capsomeres? Approximately 2130 capsomeres are present. Okay, and protomere in TMV, each protomere or each capsomere will have how many amino acids? 158 amino acids are present in each protomere. Molecular weight of this is 17,400 Daltons molecular weight. Promoters, they form a groove that allows nucleic acid to fit. The RNA of TMV is what? Single stranded, and RNA here it is single stranded, it contains 6,200 nucleotides. Okay, it contains 6,200 nucleotides. Influenza virus genome is enclosed in thin flexible helical capsid and it is folded within the envelope. So here what all are the things that you have to remember about the structure of TMV, its length, diameter, and what is the diameter of the hollow tube here, how much amount of capsomeres are present and how much amount of nucleotides are present in the genetic material RNA. If you take a ratio between RNA, between the nucleotides, amount of nucleotides present in the RNA and amount of capsomeres present in the capsid, if you take a ratio, you can see 6,200 nucleotides here, right? 6,200 is to 2,130 protomeres. It is around 3:1 ratio. Okay, the ratio is 3:1. Fine, right? Clear? It becomes vice versa when you keep nucleotides here and protein coat here, protomeres here. If protomere is first, protomere is to amount of nucleotides. If in this case it can be 1:3. Clear, right? It has started just 15 minutes, ma. So far it is 15 minutes. You can continue, ma. You can continue, and if you are having at any point if you are facing any difficulty, please drop a message in the chat box. Yeah, now. Due to some technical issue, the class is preponed. The timings of the classes is preponed today. Okay. Usually it will be at 4:30, but today it is preponed, ma. Yeah, please continue with the class. Okay. Polyhedral or icosahedral viruses. Capsids in these viruses are polyhedral with icosahedron symmetry. The symmetry is what? Icosahedron symmetry. Regular polyhedral shape with 12 equilateral triangular faces. Okay, it has 12 equilateral triangular faces, and how many vertices? 12 vertices. Capsomere count, like how you have seen the capsomere count in TMB 2130 capsomere count. That count will not be seen in the polyhedral or icosahedral viruses. The capsomere count will vary. Okay, it can be 12 up to 812 also. 25 capsomeres are found in adenoviruses. 25 capsomeres. Each capsomere, amongst these 25 capsomeres, each capsomere will have 5 to 6 protomers. Each capsomere will have 5 to 6 protomers, and pentamere, pentons, which is made up of 5 subunits located at the vertices. Hexameres, hexons, it has how many subunits? 6 subunits. The name itself says, right? This forms edges and triangular faces. Fine, right? Next. Complex viruses or tadpole structure. Here if we go through bacteriophage viruses, they have complex capsids. They consist of polyhedral head and a helical tail. This structural form a binal symmetry. Okay, what type of symmetry is seen over here? Binal symmetry. They are neither purely icosahedral nor purely helical. Okay, they give us a tadpole structure with a capsid head, and a collar attached to the long sheath, followed by tail and tail fibers. Tail fibers at the end of the tail fibers in the bacteriophage, they have what enzyme? Lysozyme enzyme, which will degrade the wall layer of bacteria. So here bacteriophages may possess single or double stranded DNA or RNA. Examples if you see T even phases and T odd phases. These phases will infect what bacteria? Escherichia coli. T4 phase is tadpole shaped. It has 5 structures: head, head tail connector, tail plate or base plate, and fibers. Fine. Next. Head, elongated bipyramidal or hexagonal prism, here you can see. Fine, right? And a long tail. It has a connector, connector is collar, which connects to the head and tail. Fine, right? Next, the sheath, complete sheath here, inner tubular core sheath, it has how many protein subunits, ma? 144 protein subunits in 24 helical rings. Here you can see. Okay, 144 protein subunits in how many helical rings? 24 helical rings. Fine, right? The sheath connects head on the one end and base plate on the other end. Here fibers will originate from base plate points. T even polyphases exhibit thin structures over here. Clear? Fine. And then viral genomes. Here it is about viral genomes. As we have discussed, it can have single stranded or double stranded DNA, but any one of the types. All 4 types are found in animal viruses, but mostly it is DNA. Whereas plant viruses will only have single stranded RNA. Bacterial viruses, generally they will have double stranded DNA. Okay. Generally bacterial viruses will have double stranded DNA. Smallest genome is having 4000 nucleotides. They can code for 3 to 4 proteins. Examples of such viruses which has smallest genome can be MS2 and QB virus or QB bacteriophage. Here you can see DNA you have in 2 types, icosahedral viruses and helical viruses. Naked DNA you can see in adenoviridae. Fine. Enveloped viruses you can see herpes viridae is one example. Helical, pox virus. Fine, right? Any doubts here? Yes, you are right. Pi 174 phase icosahedral, first DNA sequenced by it is Sanger. Most important point. Good, ma. Next. Yeah. Virus like bacteriophages, herpes virus and vaccinia virus, they have genomes ranging from what? 1 to 2 into 10 to the power of 5 nucleotides. This code for over 100 proteins. Fine, right? Viral genomes can be linear or circular, single or double stranded as well. Among RNA viruses also, they have double stranded RNA genomes, like they will infect animals, plants, fungi and some bacteria as well. Most of the viruses will have single stranded RNA. Viral RNA is identical to viral mRNA. Fine, right? Plus strand or positive strand it has. For example, you see polio, TMV, Brome mosaic virus. This can begin with protein synthesis directly upon entering into the host cell. If RNA is complementary to RNA viral, then it is addressed as what? Minus strand or negative strand. Best examples for minus or negative strand you can see rabies, mumps and influenza. Okay. Many RNA viruses have segmented genomes, around 10 to 12 segments, and each segment may code for single protein. Clear? Next. Replication or viral reproduction also you can address it as. There are 6 stages for viral replication once it enters into the host cell, and it can follow 2 cycles. One is lytic cycle which kills the host cell because of viral replication. Second is lysogenic cycle where the host cell remains alive even after division. So, what are the 6 stages of virus replication? First is adsorption. Virus gets adsorbed on the bacterial cell surface. Second is penetration. The genetic material is penetrated, genetic material of virus is penetrated into the bacterial host. Third is synthesis. Synthesis of particles of viruses happens. Fourth will be replication of the particles. Fifth, assembly of all the particles and forming it to a virion, followed by release or lysis, release of viral particles by killing the host cell. Here we go with the examples. So, first stage you see my adsorption. Both lytic and lysogenic, both cycles we will see. Yeah, in lytic cycle if you observe, first adsorption where phage will land on the surface of the bacteria, followed by it penetrates the genetic material of the virus into the bacterial genome. Here if this viral genome will replicate along with it, okay, by using complete cellular machinery of bacteria. If viral genome starts replicating, then it will replicate, it will synthesize, it will assemble, and virion particles will be released by killing the host cell. But then there is another possibility also that is learnt under lysogenic cycle, wherein the viral DNA is integrated into the bacterial DNA. Once the viral DNA is integrated into the bacterial DNA, that cell is addressed as prophase. Okay? This is addressed as prophase. Now the division or multiplication of viral genetic material happens along with bacterial genetic material, and bacterial cell will not be killed in the lysogenic cycle. After many cell divisions, the viral genome will be replicating completely like this along with bacterial genome. Here viral genome will completely replicate, viral particles will be synthesizing. This complete process happens utilizing the host cellular machinery, and at the end, killing the host, the virus particles will be released. Fine, right? This is the entire process. Next, first is what? Penetration or entry. Entry of virus is mechanical and enzymatic process. Lysozyme is released by tail fibers that digest the bacterial cell wall. After pinning, the tail sheath will contract. This will push the core through the cell wall. Can you see here? Next, ATP energy is used for tail contraction. They empty the fast shell and they remain outside, which is termed as ghost. Okay, the bacterial cell which is releasing out the virions, that is termed as ghost after releasing out the viral particles. Now synthesis, you see, after viral genome entry what happens? Host mRNA and protein synthesis will stop. Phase DNA takes over the host metabolism. Phase DNA synthesizes mRNA by using what? Host RNA polymerases. Host DNA is degraded totally and its nucleotides are used to synthesize viral DNA. Fine, right? This happens in synthesis. Next, replication. Capsid proteins are synthesized using what? mRNAs. mRNAs are made using what? RNA polymerase here from the host. Viral DNA act as template. Different proteins are used to form head, tail and tail fibers. Next is assembly. Phase DNA and capsid proteins, they are assembled into complete phase viruses. The nucleic acid is first inserted into which region? Head region, and tail and head will unite forming one complete virion. Such mature virions will replicate and new phases are formed inside the host. Fifth process is lysis. Then about lysogenic cycle we have already discussed from prophase. Just go through the points. If you are having doubt, just let me know. The phases that lyse the host cell through lytic cycle, these are addressed as virulent phases, and the phases which are involved in the lysogenic cycle are addressed as temperate phases. First, replication of plant viruses. Entry, release of nucleic acid, RNA synthesis, capsid synthesis, lastly assembly into virions. Plant viruses, they cannot enter the host cell on their own. They will enter via injection or ectodesmata or through vectors. Like insects will induce the viral particles or induce the genetic material through their proboscis. Release of nucleic acid, RNA synthesis and capsid synthesis, followed by assembly of all viral particles and then release. Fine, right? This is the whole concept about lytic and lysogenic cycle, which we have discussed with diagrams. That only theory is provided here. You can go through it. Now we have to remember about viral diseases, which is very important. Many a times in the exam, the questions are asked in the form of match the following. You have to remember all the diseases and the virus that is causing the disease. Virus like bacteria and fungi, they can cause diseases both in animals and plants. In infected tissues like eggs, bodies and crystals may be observed. Like common cold is by rhinovirus, influenza by influenza virus, poliomyelitis by poliovirus, yellow fever by arbo, smallpox by variola, AIDS, HIV, rabies virus, and COVID by coronavirus, that is SARS-CoV-2. Plant virus, diseases like tobacco mosaic, TMV, tomato leaf curl by tomato leaf curl virus, papaya mosaic virus, potato X virus will cause potato mild mosaic disease, citrus tristeza is caused by citrus tristeza virus. What is the symptomology of virus? Virus infection will first cause, first you see the examples like tobacco ring spots, tulip flower breaking, vein clearing in bendy, necrosis, mosaic disease in tobacco. Symptoms can be both external and internal symptoms. External symptoms include pigment discoloration, stunting, malformation, growth reduction, and tissue deterioration or necrosis. Whereas external symptoms, when they are further classified, it can be classified into mosaic, flower color breaking, yellowing and yellow vein coloring. You see pigment discoloration, flower breaking, like patchy discoloration is observed on the flowers. Best example for this is tulip flowers, which is first observed by Charles. Next is yellowing. Chlorophyll loss will lead to yellowing of the leaves. This increases carotenoid concentration and gives yellow color of the whole plant. Next is yellow vein disease. This causes discoloration of the vein. Yellow disease is observed, where rest of the leaf will be green, only the veins will be yellow. Stunted growth, overall plant leaves, fruits, they will yield in reduction. Malformation, irregular growth will happen. This is caused by hyperplasia by increasing cell number or hypertrophy by cell size increase. Both the conditions can lead to malformation, either cell number increase, that is hyperplasia, or cell size increase, that is hypertrophy. Next is necrosis. Cells surrounding the infected area will die. This is referred to as necrosis, which is typically light brown or dark brown color. Example, you see tobacco necrosis. Internal symptoms can be tissues show hypertrophy, which increase the cell size, or hyperplasia by increased cell division. Sometimes cell size will decrease drastically, they cause hypoplasia. Transmission method, seed transmission, examples like tobacco ring spot or barley stripe, which is infected by ovule mosaic virus. Pollen transmission, stone fruit ring spot virus like pollen grains. Dodder transmission and fungal transmission. Most important features here you have to note is examples in which they are transmitting and vectors. From this entire table, examples and vectors are important. Go through it and you can also take a screenshot. Control measures of viral infection, completely memory based. Go through the table carefully. Rice turgor disease, which is the type study that we have to learn, be it bacterial infection type studies that we have learnt, or now viral diseases like rice turgor disease. We have to majorly focus on the symptomology. What is the symptoms that are caused by this disease and what is the virus that is infecting. If you see the symptomology, this affects all growth stages of the plant, like stunting growth and reduced tillering was observed first, stunted growth and reduced tillering. Leaves will become yellow or orange yellow. They may develop rust colored spots. Discoloration is seen at the leaf tip. Discoloration is observed at the leaf tip, which progresses down to the blade or lower leaf. And young plants, few tillers will develop and root growth is incomplete. Delayed flowering with small and poorly exserted panicles is observed. Most panicles are sterile or they have partially filled grains. Next. Younger plants will show stunted growth, small panicles and unviable deformed seeds. Virus localization in vascular tissues, this leads to reduced soluble sugars and increased starch and amino acids. Pathogens, what are the viruses? Two complex viruses like rice turgo bacilliform virus, rice turgo spherical viruses are observed, and how they are transferred. Like vectors should be learned for sure. Vectors like leaf hoppers, most efficient vector is green leaf hopper. Fine, right? Next, what are the control measures? Control measures you can go through. Next is tobacco mosaic virus. Symptomology is important here. Mosaic patterns on the leaf, leaf malformation, yellow streaking, stunted growth and infected fruits. These are the symptoms that are observed for tobacco mosaic disease. Fine, right? Just a second. The structural description of TMB we have dealt previously only, and economic importance of bacteria. Most importantly, all the tables which I have given here, you can take screenshot of this and revise it multiple times. Each table is important, ma'am. Like what is the economic importance of bacteria? Each table is important. Important antibiotics. Of course, this we have dealt in our first class also, economic importance of bacteria. Diseases of bacteria also we have done. Now, it's questionnaire time. Fine, ready? Look at the first question. What does bacterial cells possess in eukaryotic cells? What does bacterial cells possess in eukaryotic cells? Bacterial cells doesn't possess any photosynthetic pigments. They don't have 9+2 arrangement. Membrane enclosed organelles. No, there are no membrane bound organelles in bacteria. Bacteria has a nucleoid with circular chromosome. No, it is option B, ma. Okay, they have a nucleoid with a circular chromosome. See, here hits the question. Bacteria cell, even if it is in eukaryotes after infection, or it has bacterial characteristics only, no? It has a nucleoid with circular chromosome. Next, plasmid. Plasmid. Give out the correct statement with respect to plasmids. They replicate independently of the main chromosome. Yes. Plasmid DNA will replicate independently of the genomic chromosome or genetic chromosome. Next, often they contain antibiotic resistance. Yes, plasmid DNA will give antibiotic resistance. All they are transferred from one bacterium to another by conjugation. True. So, all of the above is the answer. D. Very good, ma. Next. Go through the explanation also. Bacteria are prokaryotes, they have nucleoid. Membrane enclosed organelles are absent. N+0 microtubular arrangement is seen, not 9+2. Photosynthetic pigments occurs in some bacteria but not all bacteria. Only those bacteria which are photoautotrophs, in them photosynthetic pigments are present. Next, all of the above, like plasmids have extra chromosomal DNA, antibiotic resistance is observed, they can be transferred between bacteria via conjugation. Next, look at this question. Okay, please don't look into the answer. Yeah, now. One of the following bacteria is used as good vector in genetic engineering. Most noted. Of course, it is Agrobacterium tumefaciens, right? Agrobacterium tumefaciens, this is widely used natural genetic engineer in plants. It possesses what plasmid? Ti plasmid, which is used to transfer tDNA, good, into the plants, and this property is exploited to introduce foreign genes to the plant genomes in genetic engineering. Till here it is fine, right? Mycobacterium is pathogenic, Salmonella pathogenic, Bacillus is having industrial importance for enzymes. Next, about bacteria, bacterial cell. Of course, in bacteria, mesosome is present, right ma? Mesosome is present. Nucleoid represents the genome, true. Ribosomes, they are found in cytoplasm. Histoproteins are complexed with DNA, wrong. The incorrect statement, if we see, incorrect statement with bacteria here is histone proteins. Because histone proteins are absent, rather some positively charged proteins like polyamines will be present to hold the DNA, genomic DNA, right? Next. Correct statements will be 1, 2, 3 and wrong will be 4. Of course, I have explained you the question again. The cell wall of both bacteria and cyanobacteria will contain. Don't look at the answer, please. Have drafted for the required explanations as well, but you don't see the answer. Cell wall of bacteria and cyanobacteria. What is the common feature in them? The cell wall of bacteria and cyanobacteria. Muramic acid, right? Because muramic acid, it is a component of peptidoglycan. Both bacteria and cyanobacteria are under eubacteria. Eubacteria will contain what? Muramic acid. Next, about mycoplasma. Discoveries I told you, right? Once a time asked question. Discovered by whom? Nocard and Raux. Okay, in the year 1898. Next, gametic meiosis takes place in gametic meiosis. To answer this question, you have to be clear on the life cycles of algae. Anyhow, we will learn this in phycology. On the life cycles of algae, if you are clear, you can answer this question. You have to identify which algae here, whether red, brown or green algae, or the examples here which is having diplontic life cycle. because the one which has diplontic life cycle only undergoes gametic meiosis. If the haplontic life cycle is present, haplontic life cycle, zygotic meiosis occurs. Zygotic meiosis occurs here. Fine, right? So, sargassum is the right answer, which has diplontic life cycle. Syphilis disease, from the table itself you can answer. Treponema pallidum, right? Next, bacterial genera carrying out nitrification and nitration. First, ammonia to nitrate is by nitrosomonas. This is nitrification, and nitration, if you see, from nitrite to nitrate, that is by nitrobacter, and nitrococcus is also the example. Fine, clear till here? Next question. Good. Endosymbiotic evolution, many a times asked question about endosymbiotic evolution. This is for both semi-autonomous cell organelles. What are they? Chloroplast and mitochondria, both of them, okay, which are semi-autonomous or self-duplicating organelles. Fine. Next, first smallpox vaccine is an example for what? Smallpox vaccine. Who discovered that? Edward and Jenner, right? This is an example of live attenuated vaccine. Slimy and slippery nature of nostoc is due to what covering? Mucilage covering. Fine, right? Very simple question. Next, about 3 domains, 6 kingdom we have learnt. What are 2 different domains of prokaryotes? Archaea and bacteria. HIV virus, it has what genetic material? Single stranded RNA. Right. Next, which among the following media will actually favor the growth of certain organisms while suppressing the growth of others? It is what media? Selective or synthetic media. You can answer carefully. Is it selective or synthetic media? The right answer is synthetic medium, right ma'am? Sorry, selective medium, because in synthetic medium what happens? All the culture medium, all the bacteria which is present in that medium will be grown, whereas selective medium will support the growth of few organisms and the inhibitors present in that will inhibit the growth of the rest. Blue-green algae that causes red blooms. What type of blue-green algae is it? Trichodesmium erythraeum. There is one dinoflagellate which also causes red tides in the Red Sea, that is goniolax. Don't get confused between goniolax and trichodesmium, okay? Next question is on exotoxin. Produced by Clostridium botulinum. Is by what infection? Lysogenic conversion it is. Which of the following diseases are caused by bacteria? If you look into all of this, measles, viral disease, tetanus, they are viral diseases. So here if you see in the fourth option, typhoid is caused by Salmonella typhi. Pneumonia, Streptococcus pneumoniae, tuberculosis, Mycobacterium tuberculosis. So all these three are what? Bacterial infections only. Next. What is the mechanism of white blood cell which is used to kill bacteria, fungi and other invading organisms? That is through oxidative activity, where phagocytic WBCs like neutrophils and macrophages, they will kill the pathogens via respiratory burst by generating what? ROS species, those are reactive oxygen species. This mechanism is an oxidative mechanism. Next, yeah, this question. Herpes, adeno and pox virus, all of them has double stranded DNA. Parvovirus has single stranded, clear? Yeah. Answer this question. This we have learnt in our PDF as well. Which is sequenced first? Haemophilus influenza. Last question of today's session. Parvovirus will cause infection in dogs, ma. Yeah, here we go. All the following may be methods of inhibition of microbial growth by antibiotics except, okay. Except which process? All of them are what? Anti-inhibition process for microbial growth. Antibiotics will disrupt cell wall synthesis, yes. It will interfere with cell membrane functioning, true. Antibiotics will inhibit the synthesis of proteins, also correct. Antibiotics prevents release of the energy of ATP. Nowhere antibiotic will show any of the effect in the energy release, okay. Either they will show their effect on the cellular metabolism or the cell wall, but not on the energy release, remember that. So, with this we have ended up the objective practice as well. For more objective sessions, I will be doing like in short videos and in shorts, like you can get exposed to more of the questions from CUET-PG, fine, right. So, for today's session, this is the complete thing from microbial diversity. Next class we are going to start on phycology, okay. Yeah. Timing of the class will be 4:30 only, ma. If at all due to any technical issues, if the time gets preponed or postponed, it will be informed, fine, right.